In this video, we will be talking about the release of insulin, how basically insulin released from the beta cell. So this is a beta cell, and this is blood vessel. So basically, in the beta cell, this end, which is toward the blood vessel, it's called the basolateral end. This is the basolateral side, whereas the end which is away from the blood vessel, this end, it's called the apical side. Apical side. So in the basolateral side, we have two channels, this one and this one. And in the apical side, we have one channel. This one is called as ATP sensitive potassium channel. Why we call it ATP sensitive, we will be talking about this. And this is the GLUT2, and this is voltage sensitive calcium channel. This is calcium channel. So basically, GLUT2 is the channel which is towards the basolateral side, and it is a glucose sensor. So what does it do? That whenever there is increase of glucose in the blood vessel, when the glucose level increases, it will sense the increased glucose, and it will facilitate the diffusion of glucose from blood vessel into the beta cell, leading to increase of glucose in the beta cell. So there will be increased glucose. Now potassium channel, normally what it does is, when it uh, like throw kind of throws the potassium into the blood vessel, keeping the resting membrane potential of beta cell, resting membrane potential of beta cell minus 90 millivolt. So when there is increase of glucose in the body, there will be GLUT2. It will sense the increased glucose in the body. It will pull this glucose into the cell, and this glucose, it will get metabolized and will lead to more ATP production, and this ATP will inhibit this potassium channel. That's why we call it ATP sensitive. ATP sensitive potassium channel, because it depends upon the ATP level. When the ATP level are less, it will keep on working. When the ATP level gets increased, it will inhibit this channel. So when the potassium channel gets inhibited, this will cause increase of potassium ions in the beta cell. So there will be more positive charge that will bring the resting membrane potential, the membrane potential, to minus 40 millivolt. Now this calcium ion, just like potassium channel, it is ATP sensitive. This calcium channel is voltage sensitive. So it is voltage sensitive. It is voltage sensitive. It is voltage sensitive. So whenever the voltage reaches to minus 40, it will stimulate the calcium ion and will cause more of calcium ion into the beta cell. So basically, for insulin release, we need three factors are needed. What are those three factors? First is more of ATP. We have more of ATP. Second is uh, more of calcium ions. We have more of calcium ions in the cells. And the third one is a protein called as snare protein. This snare protein is present here, somewhere here in the apical side. So these three factors, that is more of it, this increased ATP, calcium ions, and the snare protein. This will facilitate the release. How? Here in this side we have this vesicle having insulin. So it will these three factors will facilitate the release of insulin via exocytosis. This will cause release of insulin into the body. So these are the basic steps for insulin release. So basically we can say that we have, like, there are four important steps for the insulin release. The first one is this GLUT2, which is a glucose sensor. The second one is closure of this ATP sensitive potassium channels, and the third step is the this one, that is uh, this third one is the opening of this voltage sensitive calcium ions, and the fourth one is this exocytosis by insulin. So these are the four steps which are required for insulin release. And now some important conditions related to insulin like diabetes mellitus. It is a condition where there is excess of glucose, so we need insulin to convert this glucose into glycogen and decrease this glucose level basically. So to decrease this glucose level, we have a drug called as sulfonyl urease. So basically this potassium ion channel, it is also called as SUR. SUR stands for sulfonyl urea receptor. So this SUR, it's sulfonyl urease. It is a, we can say it is a insulin secretagogue. Insulin secretagogue. So what its action is just like ATP. Just like ATP, it inhibits the potassium channel. So just like that, it also inhibits the this potassium channel. By inhibiting the potassium channel, we all know that this will lead to release of insulin. So more insulin will be there, which will convert this excess, which will decrease this excess of glucose. Second condition is insulinoma. Insulinoma. So this insulinoma is a condition where there is excess of insulin. So for that we have a drug called as diazoxide. So what it will do is it will open the potassium channels. It will stimulate or open potassium channels. So we all know that when this potassium channel will open, when this will be open, this will lead to more of potassium that will lead to more excess of potassium will be released into the blood vessels. So when the potassium is released into the blood vessel, this resting membrane potential will remain at minus 90 millivolt. This will not facilitate this calcium ion opening and all. So this will inhibit the insulin release. So basically it inhibits the insulin release. This will lead to decreased insulin, so which will be helpful in condition where there is excess of insulin, that is insulin. So basically this is all about release of insulin.